Yeah, so we've been doing experiments in space since 2019, and so far we completed 8 missions. And most of those missions are done in a semi-autonomous fashion, meaning that we have a payload that contains our brain organoids in there, and the experiments are autonomous inside. So the only request from the astronaut is to plug the payload into a power outlet so we can have access to all the information that runs in there. And during these 8 missions, we were able to accomplish a lot, and mostly understanding the fundamental, basic molecular, cellular alterations that happens in these organoids upon exposure to space. But the experiments that we want to pursue now are way more complex, and there is no way or no time to train a professional astronaut to perform these experiments. So the best experiment would be performed by a scientist. So the idea is to train a scientist, in this case me, and volunteer myself, to become an astronaut and perform these experiments. So I'm being driven by the science. The science is requesting to be performed in space. So we go where the science is asking us to do it. We have the first drug discovered in space for a neurological condition that is entering clinical trial here on Earth. And this is a drug that we found out for Rett syndrome, which is a severe neurological condition. So it's a mix of serendipity and accidental. One of these missions, the organoids that were supposed to go got contaminated. And we had these extra organoids from Rett syndrome, derived from people from Rett syndrome. And we sent them to space in one of the payloads. And what we learned was very interesting. These organoids from Rett syndrome show high levels of endogenous retrovirus that are being activated in space. So these endogenous retrovirus are normally suppressed for healthy people here on Earth. But as you age, they get released into your brain. And this is something that every single cell contains. So everybody have these endogenous retroviruses, but again, most of the time they are silenced through life. In Rett syndrome, what we figured out is that the protein that is mutated in Rett syndrome controls the activity of these retroelements. So it got completely de-silenced in space, creating like a pro-inflammatory response. So the brain cells who actually accumulated this retrovirus are thinking that they're being attacked by a virus, but they're not. This is all endogenous retrovirus that are being accumulating into the cells. And by creating this accumulation, it triggers this inflammation that is detrimental to the neurons. So what we're learning is if we want to reduce the inflammation, we need to use antiretroviruses to treat a neurological condition. And seems counterintuitive. Why are you going to use an antiretrovirus to treat a brain disorder? But actually, the space project revealed that that makes sense. So we did all the preclinical studies using brain organoids here on Earth, as well as mouse models, and the drug seems to be quite effective. So we are moving now into a clinical trial to test that in people. Penicillin, for example, was discovered by accident, and was the human eye who actually detected that, and was the human mind who interpreted the experiment and realized that penicillin was being helpful to kill the bacteria, right? So a robot will never discover penicillin. And then he was able to observe this halo where the bacteria couldn't go. And this was because the fungus was producing some kind of a substance that was inhibiting the bacteria to grow. And there are also other examples in science where serendipity and the curious observation of the experiment was able to bring huge insights into science. So one example that I like a lot is the discovery of photolysis. So this is like a DNA repair enzyme in bacteria. And so there was like conflicting results. So one example that I like a lot is the discovery of photolysis. So this is like a DNA repair enzyme in bacteria. And so there was like conflicting results. People were irradiating the bacteria to cause DNA damage and looking for the cell death of these bacteria cells. And some of the bacteria will die. But when he would repeat the experiments during the day, the bacteria will survive. So we realize that if you are close to a window or you have natural light, that will activate photolysis, which will act to correct the DNA. But during the night, there is no light to activate the photolysis, so the cells will die. So we have most of the equipment ready to go, maybe like small adjustments here and there for the microgravity stuff. But we have a list of experiments to perform during the flight, during this 3-4 days flight, and we can accomplish a lot, especially because I'll do it myself. I think one of the most interesting experiments that might be translatable to people immediately is to test neuroactive compounds. They are actually curated from the Amazonian biodiversity. So these are molecules that comes from plants where the original tribes, the shamans used to use to cure people from different disorders. So we are curating those molecules and we're going to be testing them as a neuroprotector in brain organoids in space. So this will require the use of brain organoids derived from different disorders such as Alzheimer's, Parkinson, age-related disorders, as well as neurodevelopmental conditions such as autism. The way to have organoids that mimics symptoms of Parkinson or Alzheimer's is to reprogram cells directly from the patient. So we actually get cells, live cells from people with Alzheimer's, let's say. We reprogram those cells back into an embryonic stage, and from that embryonic stage we can make brain organoids because they capture the genome of the person, and these are highly genetic conditions. The genetic dictates some of the alterations that we see in those brain cells. So we can actually recapitulate by creating an avatar of a brain cell of that person in the lab. So this technology can also be used as a diagnostic tool. For example, I could get cells from people who are young and normal, neurotypical, with no genetic condition, and I can age these cells in space to see if some of these people will actually develop any form of dementia or Alzheimer later in life. So we are actually hopeful that this can also be used as a diagnostic tool. In the case of Parkinson, we can reprogram cells from people carrying genes that are mutated in Parkinson disorders, and then we can recreate specific brain regions that contains dopaminergic neurons, and we can see if they fail on producing dopamine. We actually have done that in the lab, and they reproduce quite well some of these alterations that we see in the Parkinsonian brain. So another use is brain cancer, such as glioblastoma. And that is very interesting because what we do is we use a normal, a neurotypical brain organoid, and we seed the patient cells, the patient glioma cells inside of these organoids. And we see little bumps of cancer being produced inside these brain organoids. And then we can test different drugs to see which ones can eliminate the cancer cells while remaining the brain cells intact. We've done this on ground. We know that we can do it. But in space, we have the ability to accelerate the entire process. So everything, the drug discovery can be done faster than on ground. The beauty of this strategy is that can be personalized. So we can generate cells and brain organoids or other brain tissues exactly from the cells of the person. So we capture the genetic background of each person. And we can test. Sometimes some drugs might be useful for one person, but might be toxic to another one. And we can detect that in vitro before you actually test into the patients. So we actually plan to work with a variety of genetic backgrounds, including myself. I'm putting myself on the experiments as well. So that opens this idea of personalized medicine. Space exposure affects the astronaut's brain. So we have lots of evidence that affects human cognition. And I think one of the best experiments or examples is the NASA Twins experiment or project, where you can see like the decline in cognition in the astronaut even after 6 months upon return to Earth. This is like a big worry for long-term space flight. It could be even a problem for tourism in space or human colonization down the road. And so one experiment that we're planning to do is to measure the electrical activity of brain organoids during space flight to understand better how the networks rearrange in the cortex while in microgravity. So that might give us some cues on first how to protect, how to eliminate, and even what are the bases, why this is happening. Is it really because of microgravity, or this has to do with cosmic radiation? So we are planning to perform some experiments to isolate some of these factors so we can be more sure of what is really the cause of those alterations. Two of the major factors that might be contributing to these alterations at the cellular level is the microgravity per se, and we have tons of evidence that this might be a problem, but there is also like cosmic radiation, right? So one of the experiments would be, what if we send organoids that are protective against cosmic radiation? So how to do that? So there is a gene called DSUP that is present in tardigrades. Tardigrades are these small bears, microscopic bears, that survive drastic conditions. They can even survive cosmic radiation because they can be found on the moon, they can be found outside the space station. And this is because they express this gene called DSUP that basically suppress the cosmic radiation. So we engineer human brain organoids to carry this tardigrade gene now. So these transgenic brain organoids would be sent to the space station to see if we can recapitulate some of the senescence observations that we have. So if we continue to see the alterations in the brain cells, we can claim that this is due to microgravity, no longer cosmic radiation, because we isolate that factor. So if everything goes well, I'm planning to go to space to perform the experiments myself. And hopefully we can do that by the end of this year, 2025. And the idea was to go with a crew on a SpaceX flight and perform these experiments in orbit inside the Dragon capsule.